All right, so now we're going to go over the second part of Chapter 6. In this part of the chapter, we're going to go over 3 different structure determination techniques, and those techniques are circular dichroism, nuclear magnetic resonance, and X-ray crystallography. So we've already talked about one structure determination technique in previous chapters, which was Edman degradation. Edman degradation helped us determine the primary structure of a protein, or just the sequence of amino acids in a polypeptide. These next 3 methods that we're going to discuss today are going to be more about the shape of the protein, so the secondary, tertiary, and quaternary structure. So first we'll learn about circular dichroism. Circular dichroism tells us the secondary structure specifically, and then nuclear magnetic resonance and X-ray crystallography will both give you more global information about the tertiary and quaternary structure, so the overall protein shape. So let's go into each of those individually. So first we'll talk about circular dichroism. So as we should know that when you have circularly polarized light passing through chiral molecules, chiral molecules will absorb different direction of circularly polarized light differently. So if it's circularly polarized to the left or to the right, it will look different once it passes through a chiral molecule. And we already know that not only are individual amino acids chiral, the secondary structures within a protein also have directionality, right? We talked about our alpha helix having that right-handed directionality. We also know our beta sheets can be parallel and anti-parallel. And so if you look at a circular dichroism diagram, we'll be able to actually detect the secondary structure character of a larger protein. So if you look at various wavelengths of that circularly polarized light, you'll see different patterns for those different shapes. So the first shape we'll consider is the alpha helix, which is drawn in sort of yellow on this chart. And so that when you have a protein that has mostly alpha helix character to it, and you measure a CD, you will see minima right here around 208 nanometers, and then again around 220 for an alpha helix. And that's different than if it looks like a beta sheet. For a beta sheet, we'll actually just see more of the singular minimum right around 215 nanometers. And that's very different from if we had a random coil where we'd see this minimum right around 198 nanometers. And so something to sort of keep in mind is before we've talked about looking at spectra, we've talked about looking for maxima over here. For circular dichroism, we're not looking at these maxima. That can't really help us differentiate that well between the different types of structures that we can have. But looking at the minima is extremely helpful because the minima are all located at different wavelengths for different secondary structures. And so if you have a protein that's almost all alpha helix, you'll see 2 very nice minima right around 208 and 220. If you have a protein that's all beta sheet, you'll have that 1 very nice minimum right around 215. And if you have a combination, then you would end up getting a graph that looks sort of like the mathematical average of the 2. So if you have half alpha helix and half beta sheet, you'd have a curve that sort of looks like the average of the blue one and the yellow one that we see here. And so circular dichroism is really helpful for when we want to know the types of secondary structure that are present within a protein. Circular dichroism does not tell us the tertiary structure, like how those secondary structures are interacting with one another. So really circular dichroism is telling us more about the backbone interactions of a polypeptide chain, because remember, those backbone interactions are what forms the secondary structures. So that brings us to the other 2 types of structure determination that we'll discuss today that do tell you more about the tertiary and quaternary structure. So the first one we'll discuss is NMR, or nuclear magnetic resonance. So nuclear magnetic resonance is a really powerful technique that helps to provide the structure of a dynamic protein when it's in solution. So as you can imagine, proteins, when they're in solution, look kind of like in this image on the left. We have them kind of, all the different protons are pointing in all different directions. There's no alignment. They're sort of, floating around in space. They form some kind of stable conformation, but they're not acting together in any way. Nuclear magnetic resonance uses a very powerful magnet to help align the spins of all of the hydrogens in a molecule. And so when it does that, you can, when you apply a field and then release it, you can sort of see how the decay of how those spins return back to their original location, and you can use that to determine the structure of a molecule. And so the decay rate that we see depends on the local environment of each particular hydrogen atom, and so that's why you can get a number of peaks. This picture on the left shows us the NMR spectrum for a fairly simple small molecule, and it can be fairly easy to figure out what molecule you have based on this NMR spectrum. On the other hand, when you have a protein, it looks more like on the right. It's a really big molecule. You have a bunch of different hydrogens in it. They're all, doing their own thing. And so this kind of, for lack of a better word, messy diagram is not going to help us, this one-dimensional NMR. So when we want to figure out the structure of proteins, we will use multi-dimensional NMR. And one technique that we'll frequently see is called NOESY, which stands for Nuclear Over-Hauser Effect Spectroscopy. So that's NOE equals Nuclear Over-Hauser Effect. And so basically what this gives us is a two-dimensional spectrum. And so that allows us to go into more finer detail when we're looking at those decay rates by adding on additional magnetic pulses. So you might have your one main pulse that you allow to decay, but then you'll add in additional magnetic pulses. And by observing the combination of the different decays that you'll see, that allows you to put together this two-dimensional graph that gives you more information about the interactions of the different protons within a protein structure. And so, so let's go into some examples here, actually. So here we have along this axis, or this line where you have a slope of 1, essentially, that's going to show you the regular order. That's like the original one-dimensional diagram, and then off of that you can measure these angles, or these interactions between the different parts of that singular line. So if you look on the left here, we have this little line labeled N to C, right? That's the order of some of the protons in our protein. And we can see how they interact with each other based on this NOESY spectrum. So if we look at proton A, which is, let me get a different color here. So if we look at proton A, that's located over here. And then if we look at proton D, that's located down here. And we look for the interactions between those two, the cross peak, to see if there's a mark on the spectrum there, then we know that those two protons are interacting with one another. So here, if we connect them together, we can see right at point I on this diagram, proton A and proton D are interacting with one another. So those two protons must be near each other in space. And so by using this pattern of all the cross peaks and how strongly they're interacting with one another, which ones are interacting and which ones are not, you can start to sort of build a potential structure. And you can average together results from several different experiments to sort of figure out what structures are the most likely and what structures are the least likely. So here in this picture, we have the average of multiple different NMR NOESY spectra. And we can see, for example, over here, this looks pretty much like a beta sheet, right? We have these kind of parallel strands that are pretty parallel to one another, and they're connected by what appears to be loops, which I've circled here. And the loops you can see are a little bit more variable in their shape and their exact location, right? You see a number of different lines there. They don't overlap quite as precisely as the actual strands that are within those beta sheets. We can even start to look at some of the different types of side chains that we might see, which are shown here in these different colors. And those type of side chains will help determine, of course, interactions by hydrogen bonding between different side chains. And so, again, you can look at hydrogens there to see what's close in space to what. So, given all of that, let's talk a little bit more about how to identify the pairs of protons that are interacting with each other in NOESY. So the NOESY has the ability to identify protons that are within 5 angstroms of one another. So if they're further away than 5 angstroms, you won't have that cross peak. If they're 5 angstroms or closer, they will have that cross peak. And so using that information is really all you essentially need if you're a computer to ultimately figure out the three-dimensional structure of this protein. So we already looked at our first example. So with proton A over here and proton D over there, they came together at point I, and so those residues are 4 amino acids apart, right, from A to D. So that might give you this sort of loop structure from A to D at point I, which is the interaction we're seeing where the protons are close to one another. If we look at our next example down, we have a different sort of hairpin structure where we have proton C over here also interacting with proton D, and they come together here at point J, and that structure might look something like this, where you have this sort of interaction. And then our last example here is going to be between proton C and proton B, which is up here, and they come together at point K, which is here. So we might be seeing something like this, where the two of them interact. And so by sort of averaging together all of this information you have about which protons are close and which protons are far, and their arrangement linearly, you can ultimately identify a structure with fairly good resolution. And so just to really sort of simplify this, because we're not computers, here's a sort of idealized NOESY spectrum that gives us just the interaction between these 5 particular protons. And so those are numbered, again, along our straight line here. It's our sort of slope of 1. We have our 5 different protons. And then to look at the interactions between them, again, we look for those cross peaks. So here we can see clearly we have an intersection of protons 2 and 5 at this red spot, and you can do that in either direction. You can also go this way. You have this, and those both represent the same interaction between proton 2 and proton 5. And you can see here that they're close together in space, right? They're within 5 angstroms of one another. And that's in opposition to the other 3 protons. Protons 1, 3, and 4 are further away. They're more than 5 angstroms away from both one another and from all the other protons that are in there. So only protons 2 and 5 are close. All the other protons are far away from one another in this example. So that brings us to our next protein structure method determination, and that is X-ray crystallography. So X-ray crystallography is a really great technique that can provide the structure of a static protein that's been formed into a crystal. And honestly, one of the biggest challenges of X-ray crystallography is actually making the protein crystals. It's fairly easy to get, something like table salt to form into those nice orderly sodium chloride crystals because they're sort of small and not complex. But you can imagine, since a protein is such a large complex molecule, it can be relatively challenging to get it to form into nice orderly repetitive crystal structures. But ultimately, once you're able to do that, you are able to then shine X-rays at that protein crystal and then collect a diffraction pattern. And so the diffraction pattern represents where the X-rays are diffracted after they hit the different electrons, essentially, or electron clouds within the protein crystal. And so the pattern that you see will indicate sort of the arrangement of where there's electron cloud density and where there's less density. And that can help you to build overall a picture of what the protein might look like. And but in order to do that, to go from here's some electron density to here's what your protein might look like, you will often need to do some math called a Fourier transform. And the Fourier transform can help you construct an electron density map. And so we have an example of that on the right over here. And so once you have an electron density map, if you already have an idea of what the primary structure of the protein is, it's actually fairly straightforward to see here. It's very high quality, so you can really see where all the side chains fit in, where the backbone fits, and you can really see resolution down to practically the single atom. So this is extremely high resolution, and it tells you a lot of detail about the exact structure and shape of your protein of interest. And so so we were just looking at this map that had this really high quality, high resolution structure. But something to keep in mind is that the quality of the structure you get from your X-ray crystallography is determined by the resolution of your machinery. All right, so so something with 6 angstrom resolution, this is low resolution, might tell us maybe we have some sort of circular shape. It tells you there's an area of high electron density and an area of low electron density, but not really a lot of information about this. What we can see must be an aromatic ring, right? But as we go up in resolution, if we look at the higher resolution picture, high resolution, you can really see these distinct areas of electron density at each of those atoms, as well as the space in between. There's very low electron density, and so between your own sort of model of what you know, other parts of the structure of the protein, as well as the actual resolution of your X-ray crystallography, the quality of the structure you get can vary a lot. It can be very precise, or it can be relatively imprecise. So one example of that is shown here. So over time, people were trying to figure out the 50S subunit of the ribosome. And so on the left, we have our 9 angstrom resolution, which was one of the first ones determined. And that's fairly low resolution, right? We see a lot of fuzziness in the structure, but we still get an overall shape. We can still see maybe different domains of this protein, but we don't see a lot of detail about what the bits of secondary structure might look like inside this overall sort of tertiary shape, right? And that's at this low resolution. As we go over towards the right in this photograph, we get to this higher resolution. So all the way at the right, we have this 2.4 angstrom resolution. And you can see here we have a lot of detail. You can see all these alpha helices, you can see these loop structures that are more unstructured. There's a lot more detail in here. So that's something to sort of keep in mind as you're thinking about X-ray crystallography. So one last thing I wanted to mention is that X-ray crystallography is obviously pretty time intensive, right? You need to spend time making that crystal. NMR can be relatively quick, although maybe your computation time might take longer to actually determine that full structure. But something like circular dichroism, that first method that we discussed that tells you the secondary structure, that's extremely quick. So let's go back and look at that secondary circular dichroism. So circular dichroism can be done pretty quickly, and so that's something that you can actually use to sort of monitor a protein in real time. So for example, if you're slowly adding a component to a protein, or perhaps changing the buffer to a different pH, you can monitor in real time if there are any changes to the secondary structure and nature of a protein. So so for example, if you're slowly unfolding a protein or making it less soluble for any reason, or maybe you added a substrate and so the protein or an inhibitor or any number of things, you can monitor if there's any change in the alpha helix, beta sheet, or random coil character very, very quickly and straightforwardly using circular dichroism. So that's important to know as a technique that it is relatively quick. So anyway, with that digression, you are now ready to answer the structure determination techniques practice question.